Good afternoon, biology students. Today's Screencastify is going to focus on a phenomenal website. Actually, it's a combination of two sites related to one another. It's a DNA database used worldwide by scientists, and you're going to use it to gather information about how closely related your organism is compared to the three other animals that you have chosen to compare yours to over the past couple of days. By this point, you would have gone through homologous structures, analogous structures, vestigial structures, how they serve purposes or no longer needed, etc. You've done some anatomy comparisons. You've done some body systems, organ systems, which takes us back all the way to the hierarchical systems we talked about early in the year, and leading us up to the video you watched on a previous lesson. And now we are getting ready in today's lesson, which you'll be working through as I'm making this a day early. You'll be working through this tomorrow. Actually, take that back. You'll be working through it on Wednesday. You'll be looking at embryos, how different organisms all look pretty similar early on. And then over time, they differentiate based on the DNA, which is another reason why we're going to focus on how small differences in DNA can lead to many, many, many species. You also will be watching a little 2-minute video on how to put organisms on a family tree. This is all part of the assignment that goes with the DNA assignment. It has 5 short parts. The DNA is the most techy of the bunch, so that's why I'm making the screencast for it. But this I just want to touch on quickly. If I'm looking at, let's pretend I had a jawless fish. The jawless fish here has no jaws because obviously it says jawless, but more importantly, jaws didn't happen until the next branch up. So what can we tell about this jawless fish is it only had vertebrae. Okay, if I jump ahead to lobe fin fish, which again were fish that could start living partially on land because they had weakly developed lungs, they have no limbs, right? They're still lobes flipping around the sand, but they do have lungs, they do have bony skeletons, they do have jaws, and they do have vertebrae. That's one of the keys when you're comparing your animal to others. Anything below a category is shared or contained by the animal within that branch. So the horse basically has all this stuff, whereas the tunicates, they don't have anything. The lancelets don't have anything. Only when we get these jawless fish, they start having these characteristics. Now, this stuff does have characteristics. They're kind of at this point here, all organisms were still kind of the same, and they branched off to here, they branched off to here, and eventually to here. So onto the Screencastify focus. Here is what looks like a really intense set of instructions, but I will walk you through each of these steps. You're going to search an NCBI database. You're going to blast the DNA, where it'll bring you back to, as you see here, I picked my organism, so I'm going to use a couple of those. I listed them by common name here. I'm going to suggest you list yours by scientific name on OneZoom. Otherwise, it's going to give you nightmares and maybe a headache or two trying to sort through the information. So I think the scientific name, although it's an extra step at the beginning, will be the way to go. So let me go to OneZoom quickly. I will type in my emperor penguin, and we can see here the emperor penguin has a scientific name, so I can copy its scientific name. Control C is a shortcut. I can then go to the NCBI database and open up the link. And here is where you have to start clicking very specific things. In the instruction, it says use the database. It says gene. It tells you to type in cytochrome C plus your animal name. And again, scientific name works great. I can use my common name because I know what I'm looking for. But when I was searching some of your animals, it was a lot of stuff happening. So for instance, the tiger had a tiger mosquito, a tiger eel, and lots of again, very technical thing. So I want you to get there in the simplest way possible. So I'm going to now paste my penguin scientific name, and I'm going to search it up. And I and Mr. Novick have included a document for you to enter your data, very similar to the table I have in my slides. So it says you're going to look for a code called COX2. Any of these COX codes, again, go with cytochrome C. Basically, that's a protein that's found in the mitochondria. So again, a lot of former words from earlier this year are coming back into the picture. I'm going to use the catalog number, which again is described. It's the NC number. I'm going to, again, hold down the clicker button and drag the mouse, Control C to copy it. And then you're going to open up the next site. So I've already been to OneZoom to find my animal. And I'm now going to go to the BLAST site. And this again has a set of instructions. I'm going to enter my animal up here. This will be your sort of comparison animal, which is again the animal you've been researching. And then you're going to find 3 more animals that you're going to put in another text box that will open up. So this is another important step. You got to click line 2 or more sequences, which will give you the opportunity to compare percentages of DNA shared among the 4 animals that you've been researching. Actually, it's 3 of them compared to your own. So I'm going to click this. It's going to type in my animal name. You don't have to worry about anything extra, but notice how now it opened up a subject sequence box. And this is where you're going to do the same procedure, but you're going to type your other animals or copy and paste your other animal codes in. So if I go back to OneZoom, I know my next animal is the little penguin. So I've already predetermined these, which you have also done. So if I type in little penguin, here is the little penguin's scientific name. Again, it's a Latin name. The genus and species, the genus is capitalized, the species is lowercase. I'm going to Control C it. Once I've done that, I can ignore what I did before. And so I can X out of that. I'll go back to this site, and where'd my other window go? Here's my other window. Again, it's set up for gene underneath the catalog drop-down menu. I have to again type in cytochrome C. It looks like there's different ways you can do it. This is the way I've done it. And I'm going to paste my little penguin's scientific name. I'm going to search it up. And again, this is not super fast, but it's way faster than OneZoom. Here again, notice I don't have much to choose from because I put my scientific name in. The catalog numbers are the same for all the COX codes. I just, for whatever reason, choose COX2. I'm going to copy and paste, Control C, and I'll go back to this thing now, and I'll Control V because I'm comparing my little penguin to the emperor penguin. Now, you could at this point blast it, which is down here. So I'm going to just show you this. We're going to use this thing called somewhat similar sequences because they are generally the same type of animal. And we'll in fact use this for all of them, even if your animal gets quite different. At some point, I'm going to click this blast button, but I'm not going to do that yet because I'm going to find my other 2 animals and put them in here. In fact, I'll just do one more for simplicity. I'm going to go back to my slide quickly. And here's my little penguin. I have a crocodile, so I'm just going to jump ahead to the bottlenose dolphin. Notice here I also put in when did it branch away. That's all from OneZoom, 320 million years ago, whereas the little penguin only branched 25 million years ago. Actually, 22.8, I think, is my official number, but roughly from penguins 25. So I'm going to do the same procedure. Notice here I already have my codes in from previous years. But I want to go back through the same process. So I'll go back to OneZoom. I'm going to put the bottlenose dolphin. And notice here we have a few things to choose from. Here's kind of the overall group. So I want to keep it here. I'm going to put the common one. So you might have some options. I always stick with the ones that are most familiar. Again, Control C. I will now back out of this screen. Might have to refresh. You could always click the link each time on the slide. Again, if I wanted to, I could come back here and click the NCBI database every time, open a new tab. I could open a new tab every time here. But I wouldn't be advised because they need to be repeating steps. So ignore I even said that. So if I go back to where I left off, I'm going to, again, paste my common bottlenose dolphin scientific name. Control V, Tursiops truncatus. I'm going to search Tursiops truncatus up. Again, I'll look for the COX2 code. And here we again have a bunch of other stuff. So I'm going to look, just always make sure it says what I'm looking for. Yes, bottlenose dolphin. Here's my code. I'll copy it, Control C. I'll go back to my blast site. And again, I should put Nile crocodile in here, but I don't need to show you the same thing 3 times. Twice will be good enough, because the fun part is when you see how cool this is when it spits out numbers. It says how closely related something. So Control V, my error. I don't want to Control V just yet. I thought I copied the catalog number, but apparently not. So let me go back here and try Control C again. Now I'll try to go back here and hopefully it'll punch the number in correctly. All right, so now you'll enter your third animal. I would enter the Nile crocodile, but for sake of example, 2 will be enough. Again, make sure this is optimized for somewhat similar sequences. Again, animals are animals, but dolphins and birds are closer than dolphins and ants or beetles. So this is the safest bet. At this point, I'm now going to click blast. And as it does its magic, I'll scroll down to the bottom of the page, and you're going to use the thing that's called percent identical. So here we can see my little penguin is 89% the same as the emperor penguin, but the dolphin's only 69%. So I'm going to take those numbers. Again, you can copy and paste them or remember them. I'm going to go back and I'm going to enter them in my table. So now notice here, I actually made a separate slide and wrote out a paragraph that gave me those same numbers. You're going to have that percent identical box in the table that was created in a Google Doc that you'll be working on when you do this assignment. So here's my numbers: 89% little penguin, 69% dolphin. Here we can see my crocodile was 69.97. And here's the part that I really want you to connect. If I go back to my data, or if you went back to OneZoom and checked out the data, it makes sense that crocodiles and bottlenose dolphins are very different, 69% versus 89%, because they branched off 200, 300 million years ago, whereas penguins are only separated by 20 some million years. So it's another bit of evidence when we look at structures, how they change over time. Remember, that's always predicated on when DNA changes accumulate, things can become new species. Now, I just want to pop into the assignment sheet you'll be working on. You'll be looking at the embryo study as I showed you. Here's some more embryo stuff, and here's your DNA table. So here is where I would type emperor penguin, or I'd type emperor penguin. This is where I type emperor penguin. Here's where you put your catalog number. So if I were to go back to my site, I could get the catalog number, but because I've jumped screens now, I'm just going to go here and I'm going to get my emperor penguin's catalog number. put it into the table as you will do for your organism. And percent identical, again, that number would be 100% because it's your animal, so it can't be any different than itself. Here I had the little penguin. And I know it branched off. Again, you have to get the branching off from one zoom. That'll be something you'll determine by scrolling out as you did last week. So again, I'm going to grab my little penguins code. I see it's 25 million years ago. Actually, I know it's 22.8 if I look at a future slide, but we'll just use 25 for, again, simplicity. And I would enter that 25 million years ago that it's been separated. And while A, we'll just put 25 million. And we, again, from my research, it was 89.04% in common. And I do the same thing with the bottlenose dolphin, enter the catalog number, enter the animal name, the common name. You don't have to do the scientific name here. Common name is fine. Or you could do the scientific name and put your animal in parentheses, but just to prevent me from having to research every animal by scientific name, it's okay here if you put common name. In fact, it's recommended, but you definitely want to put scientific name on the other side. In fact, I'm going to take a moment and show you that now as to why I put scientific name where I did. So if I were to go back to here, let's just do it the simplest way. If I were to go back to my database, and again, I was messing around with this site the other day, so we got to change this one more time to gene cytochrome C, and I'm going to just put tiger. Again, we're looking for a COX number, and we're looking for what we look like a tiger, what we picture the tiger, Siberian tiger, for instance. So I'm looking here. Here's a tiger keelback. I don't even know what that is. Here's a tiger tail seahorse, a tiger snake. Here's an Amur tiger. So, hey, that sounds like a tiger. That's actually the Siberian tiger. Again, just something I found in my research. Here's COX codes, but none of them say COX2, which isn't a big deal. But what is a big deal, it says Asian tiger mosquito, black tiger shrimp, Chinese earth tiger, tiger salamander, tiger snake eel. I don't see the tiger. So if I wanted to, again, be efficient, I would go back to one zoom. And if I put the tiger name here, the first thing would usually be the most common. So the tiger we're familiar with is called Panthera tigris. If I were now to go back to, let's see if I can figure out which window was what. Back up out of here. If I were to re-enter the same data, and now I put the scientific name instead of just tiger, that's not what I want. I thought I copied it. Give me one moment to backtrack. Somehow I must not be hitting Control C properly. So let me try one more time. Panthera tigris, Control C. Too many windows open. Here we go. Control V, Panthera tigris. Now remember how messy it was before with lots of organisms that have tiger as a term in the name of the animal? Now I very specifically go to my organism, Panthera tigris. This is all the tiger that you're familiar with. Right away, I find the COX2 code. I would go over to the catalog number. I would take the catalog number, enter it in the data table on your Google Doc, enter Siberian tiger, Amur tiger, or common tiger, however you want to code it. If that's your animal, and I know some of you have tigers, it's years ago. Diverge is 0 years ago because it's every day it's continuing to be a tiger, so you don't have to, it's not separating it from itself just yet. In any case, that is about enough for me to wrap it up. There are 2 more questions that you'll look at where you actually start comparing your animal versus other animals, and this will lead you into the final week of the 2020 school year, 2019-2020 school year, where you'll be looking at what facial features, what body features organisms have and how it helps them survive in nature. So that's going to give you an introduction to that stuff. And next week, that'll be our focus. That is it for me for now. Again, this will be probably 15 minutes plus long. The main ideas are described with how to use the NCBI site and the BLAST information. Hopefully you're familiar with OneZoom. You've used it a few times. Have fun with this. It's, again, really powerful. It's what real scientists do. You're a scientist in the making, whether you know it or not. Every day is a new day and an opportunity. Make the most of it. This is Mr. Hort signing off. Have a great one.